In this video, we will discuss about how sialic acids are formed in the mammalian cells. So it is categorized into uh, three different areas where sialic acids are formed. First one is this CMP sialic acid biosynthesis that is usually occurred in the cytoplasm, and the second one is this silation part that is occurs mainly in the Golgi, and the last one is desylations that are usually occurred in the lysosomes. So we start with this UDP glic neck. So this is the carbohydrates that is bind to the UDP. It is the first converted into the man neck, and thereafter with the help of enzyme this GNE it is converted into the man neck 6-phosphate, and from this the enzymes NANS helps in the conversion of this man neck 6-phosphate to this sialic acid 9-phosphate. Now it needs to be dephosphorylated. So dephosphorylated can be done by this enzyme NANP, and this here the sialic acids are formed. However, some of the sialic acids are also recycled from the lysosomes. So in this case, what happens? The sialoglycoproteins that are found in the cells and made their final locations or final destination into the lysosomes. From there, the sialic acids are being recycled through this sialidase, that is neuraminidase, and the glycoproteins are degraded, and the sialic acids will be transferred to the into the cytoplasm through this transporter, that is SLC17A5, and from here the sialic acids are now being accumulated in the cytoplasm, and thereafter this biosynthesis of sialic acid, in the next step of biosynthesis of sialic acids, they will move into the nucleus, where with the help of this enzyme that is CMAS helps in the binding of this CMP to the sialic acids, and this compound is known as CMP sialic acid compound. However, the job is not yet done, as the sialic acid is not yet bind to the carbohydrate structures that are usually been found on the proteins, which are decorated either with N-glycosylation or O-glycosylation. In general terms, we can say that proteins that are post-translation, proteins that undergo post-translation modification might have these sialic acids. So, in order to complete this transfer of the sialic acids on the carbohydrate structures of the proteins, it needs to go into the Golgi, where the major post-translation modification occurs after the post-translation modifications begins in the endoplasmic reticulum, where the N-glycosylation begins. However, the O-glycosylation begins in the Golgi only, and it will mature within the Golgi itself. So when the CMP sialic acids move into this Golgi with the help of transporter, that is SLC35A1, thereafter the glycoproteins, that is proteins with the carbohydrate structures, will interact with the CMP sialic acid in the presence of enzymes, several enzymes which are commonly grouped as sialyl transferase. There the sialic acids will be transported to the glycoproteins, and they are formed as sialoglycoproteins. So if the sialoglycoproteins are not used further, then they will be recycled into the lysosomes. From there the sialic acids will be recycled back. So how these informations can be used? Because in the cancerous cells, particularly in the solid tumor cells such as pancreatic cancer cells, it has been observed that there is a overexpression of the sialic acids on the cancer cell surface. So because of this, the sialic acids attract a lot of attention, that it might have role towards promoting or proliferating the cancer cells. So people might have to study the impact of sialic acids on the cancerous cells. In order to study those impact, they need to somehow knock down those sialic acids. So there are two approaches that are commonly been used in the literature to knock down the sialic acids formation on the cell surface. First one is this knockdown of the CAMS gene. So that will lead to or that will prevent the CMP sialic acid formations, and thereby further sialic acids attachment to this glycoproteins will not occur. The next one is this knockdown of this SLC35A1. So in this SLC35A1, knockdown of this SLC35A1, what people have find out is that this SLC35A1 will also prevent the CMP sialic acids, will prevent the CMP sialic acid to reach into the Golgi. Thereby the post-translation modification will not be completed, or the sialic acids will not be added to the glycoproteins. And this is another way of preventing the decoration of the proteins with this sialic acids. As the proteins that undergo post-translation modification, we usually go through this Golgi apparatus, where they will move before they move to this trans-Golgi network TGN, and from there, based on their single peptide or their final destinations, they will be directed on their respective paths. So the proteins that will pass through the Golgi, the proteins that have proteins in native that have these sialic acids will have to pass through this Golgi apparatus in order to get their sialic acids during post-translation modification. So if they knock out the SLC35A1, so these sialic acids decorations will be inhibited in those kind of in those cells. So this is and then people can study the impact of cancerous sialic acid on the cancerous cells and compare them with the cancerous cells with sialic acid. So this is how these informations of sialic acids in mammalian cells can be used towards finding out some of the more insights of the cancer cell behavior that can be later used, that can or might be later used towards developing treatment, better efficient treatment against the cancers. So I hope you got a basic idea about the sialic acid mammalian cells and what is the motivations behind studying the sialic acids in mammalian cells. If you need to understand more about this sialic acid formations, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.